Hello, I'm Isabella Della Paz, and today I am presenting the article titled Sustained HIV-1 Remission After Heterozygous CCR5 Delta 32 Stem Cell Transplantation. This study focuses on a patient referred to as the second Berlin patient or patient B2 who achieved long-term HIV remission after receiving a stem cell transplant. What makes this case especially important is that the donor did not have full homozygous CCR5 delta 32 mutation that was previously thought to be necessary for HIV cure after transplantation. So these are the sections my presentation will focus on and will be broken up into. So let's begin. The background of this study is based on one of the biggest challenges in HIV treatment. Antiretroviral therapy, or ART, is very effective at suppressing HIV replication, but it does not completely remove HIV from the body. HIV can remain hidden in long-lived infected cells, which are called the viral reservoir. Because of this reservoir, most people experience viral rebound within weeks after stopping ART. Previous HIV cure cases were mostly linked to stem cell donors with two copies of the CCR5 delta 32 mutation. This mutation prevents normal CCR5 expression, and CCR5 is one of the main co-receptors HIV uses to enter cells. And over here in the image, I just have a simple little diagram of CCR5 and delta 32 and CCR5 delta 32 and how it can induce inflammation. So the main goal of this study was to determine whether long-term HIV remission could happen even when the transplanted immune cells still had functional CCR5. The authors wanted to test whether complete CCR5 resistance is truly required for remission or whether other mechanisms, especially reduction of the HIV reservoir, could explain the outcome. They also wanted to investigate possible immune mechanisms, such as natural killer cell activity and antibody-dependent cellular cytotoxicity, that may have helped eliminate HIV-infected cells. So the authors approached this goal by closely studying one patient, patient B2, living with HIV, who later developed acute myeloid leukemia. Because of the leukemia, the patient needed an allogenic stem cell transplant. The medical team first searched for a fully CCR5 delta 32 homozygous donor, but they did not find one. Instead, they selected a matched unrelated donor who was heterozygous for CCR5, meaning the donor had one normal CCR5 copy and one CCR5 delta 32 copy. The patient himself was also heterozygous. So after the transplant, the patient continued art for about 3 years and then stopped art in 2018. Figure one is one of the most important figures to include because it shows the patient's clinical follow-up over time. After stopping art, the patient maintained undetectable plasma HIV RNA for more than 6 years. This is important because most people who stop art experience viral rebound quickly. The figure also shows complete donor chimerism, meaning the donor immune cells successfully replace the patient's blood immune system. The patient's leukemia also stayed in remission, so the transplant was successful for both the cancer and the HIV outcome, as we can see in the image here. The next major results focused on the HIV reservoir. Before the transplant, the researchers detected total HIV DNA and intact proviral HIV DNA, meaning the patient did not, meaning the patient did have HIV genetic material capable of contributing to viral persistence. After the transplant, however, repeated testing showed no intact HIV DNA. They also tested blood cells, CD4 T cells, and intestinal tissue, which is important because HIV reservoirs can hide in tissues. Even after testing very large numbers of cells, they did not find replication competent virus. This suggests that the HIV reservoir was dramatically reduced and possibly even eliminated. Now, figure two addresses a major question. Was the remission simply caused by CCR5 resistance? The answer appears to be no. The researchers confirmed that the donor-derived cells were heterozygous CCR5W2 slash delta 32, as we can see here in panel A. These cells still had functional CCR5 and could still be infected in vitro by HIV in laboratory experiments. However, the heterozygous cells had lower CCR5 expression and lower susceptibility to CCR5 tropic HIV compared with fully wild type CCR5 cells. What this means is CCR5 heterozygosity may have helped, but it did not completely block HIV infection, meaning that remission was not due to complete CCR5 resistance. Figure 3 shows the patient's HIV-specific immune responses over time. After the transplant, HIV-specific antibodies gradually declined to very low or undetectable levels. HIV-specific T cell responses were also not detected after transplantation. These responses basically disappeared. This is significant because if HIV were still actively producing antigen, the immune system would likely continue showing HIV-specific responses. In contrast, immune responses to other viruses such as CMV and EBV remained detectable. This supports the idea that the loss of HIV-specific immune responses was not due to general immune failure, but instead due to the absence of ongoing HIV activity. These results suggest little or no ongoing HIV activity. Another important part of the study was the analysis of natural killer cells, or NK, and ADCC, which stands for antibody-dependent cellular cytotoxicity. ADCC is a process where antibodies help immune cells recognize and kill infected cells. Figure 4, as we can see in the image, shows the patient's NK cell populations and measures how strongly the NK cells responded during ADCC. It also shows that ADCC activity was especially strong around the time of transplantation and decreased afterward. Around the time of transplantation, the patient had very strong HIV-specific ADCC activity. The authors suggest this may have helped clear HIV-infected cells during the transplant process. They also discussed the possibility of a graft versus reservoir effect, where donor immune cells attack remaining host cells that contain HIV, similar to how graft versus leukemia effects help eliminate cancer cells. Figure 5 uses mathematical modeling to estimate how unusual this long remission is. The models in panel A, B, and C showed that maintaining viral suppression for 6 years without ART would be extremely unlikely if a meaningful reservoir were still present. The authors estimated a very low probability of remaining suppressed for this long by chance alone. Although the risk of rebound is not zero, the modeling supports the idea that the patient's biologically relevant HIV reservoir was reduced to an extremely low level. Overall, the outcome of this study is that patient B2 achieved durable HIV remission, even though the donor cells still had functional CCR5. This challenges the previous idea that only donors with 2 copies of the CCR5 delta 32 mutation can lead to long-term HIV remission. The study suggests that reservoir clearance, immune expression, and strong ADCC activity may all have contributed together as well to this remission. So why does this matter for the future? The impact on future research is important, but it also needs to be interpreted carefully. This is a single case study, and stem cell transplantation is too risky to use as a general HIV cure strategy for people who do not also need it for cancer. However, the findings help researchers understand what a cure may require. Future research can focus more on eliminating the HIV reservoir, studying graft versus reservoir effects, improving NK cell or ADCC based therapies, and understanding how partial CCR5 reduction affects HIV persistence. The main takeaway is that HIV cure research should not only focus on blocking CCR5, but also on finding safer ways to remove or deeply reduce the HIV reservoir. Alrighty, these were all of the references that I used, and thank you so much for listening to my presentation.